Welcome to the Cambridge Biology A-Level Chapter 17 Evolution. This chapter that deals with molecular evidence that reveals similarities between closely related organisms with reference to mitochondrial DNA and protein sequence data. Before going to this chapter, what we are going to do? We are going to predict the closely related organism with the help of mitochondrial DNA and protein sequences. The keywords that relate to these chapters are molecular evidence, similarities, closely related organism, mitochondrial DNA, and protein sequence data. What is meant by molecular evidence? Molecular evidence gives the details about the organism DNA, RNA, and protein, help to compare and show the relatedness among one another. What is meant by similarities? The state or fact of being similar. What is meant by closely related organism? It explains about the evolution where descendants from a few common ancestors and change to adapt to the environment where the organisms are related. In this, organisms are similar to each other but not exactly the same. What is meant by mitochondrial DNA? Although most DNA is packed in chromosome within the nucleus, mitochondria is the powerhouse of the cell, also have a small amount of their own DNA. This genetic material is known as mitochondrial DNA or mtDNA. By looking at the SNPs, single nucleotide polymorphism, only one nucleotide change, scientists can trace maternal, mother, ancestors back hundreds of years. What is meant by protein sequence data? Protein molecules are made of strings of amino acids, essentially 20 amino acids in a particular order. This string is called an amino acid sequence. Humans have shared some DNA with other organisms. If you are taking 100%, nearly 18% of the human genome, or else the DNA, is shared or similar to the baker's yeast, and 24% of his gene or DNA is shared with pine crab, 38% is for roundworm, 42% for honeybee, 47% is for fruit fly, and 65% is for chicken, 69 for platypus, 73 for zebra, 84 for dog, 85 for cow, and 88% for mouse, and 90% is for chimp. So the closest related of human is the chimp, mouse, and cow, Taurus. Mus musculus and all other chimpanzees, gorilla, orangutan will come into this category. In this chapter, we are going to see 4 examples how the molecular evidence for evolution relationship of humans with other organisms. The first example, it's none other than the variation in hemoglobin molecule of different animals while it is compared with human. For example, we are taking human and we are comparing with gorilla, gibbon, rhesus monkey, mouse, kangaroo, chicken, frog lamprey, and sea slug molluscus. And the second example is cytochrome C and its variation in different species. And the third, the biochemical variation used as an evolutionary clock. With the help of the immune reaction, we are going to identify. And the last example is mitochondrial DNA as a molecular clock, immune reaction, and the evolutionary relationship. For the first example, variation in hemoglobin molecule of different animals, what we are going to do is, we are going to the NCBI, the National Center for Biotechnological Information, where all the gene, protein, every data is present. So, what you have to do is, you have to visit the NCBI website in Google. The website is https://www.ncbi.nlm.nic.gov, where here in this you can access all databases. So here you can type any of the protein or any of the gene name, it will be directed. Here, what I'm going to do is I'm clicking the protein and I'm going to enter the protein name of interest. So type hemoglobin subunit beta Homo sapiens for human protein. So here I have typed the hemoglobin subunit beta Homo sapiens. So nearly out of 4759 list, I am going to take the second protein hemoglobin subunit beta. It's also the same, but I am going for the Homo sapiens. It gives the nearest possibilities. So click on the link. You will be directed to the particular page where you can see the data of that particular protein with the locus, that is the place in the gene, and how many amino acids it is made up of, what type of DNA, whether it linear or circular, and when it was submitted, and the definition of the protein, accession number, version of the protein. Every time it will be updated while the new sequence is submitted, and which are the, for example, Homo sapiens. This is the taxonomical order. The references are given in which paper this protein related things were referenced. So step 1, you have to check it out, number of amino acids, and the step 2, when it was updated, please go for the latest reference. And step 3, you have to scroll down to see the FASTA sequence. Either you can click here or you have to scroll down at the end of the page. I have clicked the FASTA, and this is the accession number followed by the name of the protein with the organism, and you can see the amino acids. This will make the protein. What I am going to do next is taking this protein and comparing with the other similar protein which is matching in this database. For this, a simpler tool called BLAST is very useful. So either you can separately open a new page and you can go for the base BLAST, else you can click the run BLAST. The accession number is enrolled and you have to click here. This sequence is compared with the pairwise alignment with the nearest sequences. You will give directed to this page and click BLAST. You have to click here, and the query of your sequence of interest is searched against all the sequences present in the database of protein. BLAST is still in progress. Our human hemoglobin beta subunit protein sequence is compared with all the available organism protein sequences, and it tries to find the similar sequences with the highest match. So this red point indicates that our sequence with the similarity with the other sequences. So step 1, scroll down to see the results of matching sequences. Step 2, you can see the taxonomic order of our sequence with the closest similarity with the other organisms. You can see this similar sequences matching with our sequences. So what I have done is I have went to the lineage or the taxonomic report. These are all the organisms. These are all the organisms that shows the closest similarity with the humans, and this is the score value that gives the approximate match, and number of hits also giving involved. Organism which shows the similarity with our sequences, hemoglobin beta subunit with the humans are, you can see this, Homo sapiens our sequence, Pantryloids hits, Paniscus hits, Gorilla, Pango, Hylopods, and none of this, and these are the other organisms. So I have taken the entire organism list and make it out a list in an Excel. So what I come across is these are the organisms that shows a similarity with our sequences. The first stands with the human. Our sequence is compared with the human sequence. Second is Pantryloids is a chimpanzee, and Paniscus is a chimpanzee. And third one, it's a matching with the gorilla sequence, and this is the orangutan sequence, and it gives a clear evidence that our human hemoglobin beta subunit shows the closest relationship with the monkeys and lowest score with the other animals too. So if you are scrolling down, you can see the other animals which have also the beta hemoglobin that have the matches with our humans. And the next example is the cytochrome C and its variation in different species. So cytochrome C is a component of the electron transport chain in all five kingdoms. So it's a 3D structure and function has been conserved for over 3.5 billion years. Difference between human cytochrome C with the 141 amino acid and other species are given below. So if you are seeing the human cytochrome C, the electron transport chain of chimpanzees with zero match and rhesus monkey with the one difference. That means chimpanzees and ours are similar, exactly same. Rhesus monkey only one difference, and rabbit they have 9 amino acids. The difference duck 11, rattlesnake 14, tuna 21, moth 31, and yeast 45. It gives a clear similarity that cytochrome C and variation in different species. And the next one is biochemical variation used as an evolutionary clock. That means how our immune reaction and evolution relations are same and similar. So what we are going to do is, this is a test of common. We are doing precipitin test. What we are going to do is injecting human blood serum into rabbit forms antibodies against human blood serum. When rabbit antibodies are mixed with human blood, a precipitate is formed. So, withdrawing blood from a human vein and blood is injected into the rabbit, rabbit serum sensitized to human blood is removed from rabbit. So, human blood gives a precipitin band with a sensitized rabbit serum, human blood and rabbit serum. So, you can see the differences. By comparing amount of precipitate formed between the human blood in the rabbit serum, when the anti-human antibodies react with the foreign blood serum, hints at biological readiness. The greater the reaction, the closer the supposed the relationship. So man versus man, that will be 100%. Man versus chimpanzee, 97%. Man versus baboon, 50%. Man versus dog, 0%. So, in a clear manner, how can we explain? So, human blood serum containing blood protein but no cell is injected into a rabbit. The rabbit forms antibodies that identify the human blood protein attached to them and render them harmless. So, the human blood serum is taken and injected to the rabbit, where our human antibody will be identified by the rabbit antibody and tries to attach it with them. Rabbit serum with the anti-human antibody extracted. So now a sample of the rabbit's blood is taken and the antibodies are extracted. See this? This is a precipitate that formed. So human and rabbit serum and human rabbit serum is attached with the gorilla. Rabbit serum with the baboon. Rabbit serum with the lemur. Rabbit with the rat. So decreasing recognition of anti-human antibodies to blood proteins. A sample of the rabbit's blood is taken and the antibodies are extracted. These anti-human antibodies are added to blood samples from other species to see how well they recognize the proteins in the blood of different species. The more similar the blood to human blood, the greater the reaction, which takes the form of creating a precipitate that means solid. So it gives a clear image that immune reaction and evolution relationship have very helpful identifying the differences and similarities of our related or closely related organisms. Next one is the molecular clock. What is meant by molecular clock? What is meant by mitochondrial DNA? What is genetic variation in mitochondrial DNA? What are the factors affecting molecular clock? So, before going to this, what is meant by mitochondrial DNA? Mitochondrial DNA and ribosomal RNA provides two types of molecular clock. So, different molecules have different mutation rate. Higher rate, better for studying closer related species. Lower rate, better for studying distantly related species. So, in an organism, if there is a higher rate of mutation is taking place in the sense, so we can easily identify the close related organism, if the organism have very less rate of mutation is happening in the sense, it will be very helpful identifying the distant related organism. So mitochondrial DNA is used to study closely related species. That means rate of mutation is higher. Absence of repairing enzyme compared to nuclear DNA. So what it is trying to say is mitochondrial DNA is used to study the closely related species. That means it have the higher rate of mutation is happening. Mutation rate is 10 times faster than nuclear DNA, passed down unshuffled from mother to offspring. Now you can see this picture. This is the father and mother, and these are their mitochondrial DNA. So now we can see this. This is the nuclear DNA and this is the mitochondrial DNA. So mitochondrial DNA is passed down only from mother of each generation. So it is not subject to recombination. So nuclear DNA is inherited from both the parents, making it more difficult to trace back through generation. But mitochondrial DNA is passed from only mother to mother. What is meant by molecular clock? Define. Some genes or protein sequence may accumulate mutation at relatively constant rate. That means one change per million years. That means with the help of the particular gene or proteins, the mutation rate is very less. That means only one nucleotide or only one amino acid will change in the millions of years. If the rate of change is reliable, scientists can calculate the time of divergence according to the number of differences. For example, we already know or we came from the references our human ancestors are coming from Africa, how they have identified because of this help of the gene or the proteins. So if the millions of years for a particular gene, it takes millions of years for very one particular nucleotide or one particular amino acid in the sense how many changes are happening. Based on that we can tell this much of years before the divergences happen in the group. Example, if a gene which mutated rate of one base pair has six base pair different divergence occurs in years ago, like that we can easily identify. Or this concept is called as molecular clock and is limited by a number of factors. Genetic variation in mitochondrial DNA. How the genetic variation is happening. Comparing mitochondrial DNA samples of different but related species helps to identify the divergence time period among those organisms. So by comparing the Indian mitochondrial DNA with the African mitochondrial DNA, and we can identify how many millions of years before the Indians or else the native of Indians diverged from the Africa. Like that we can check it out for the whole country or the whole world. Mitochondrial DNA changes 5 to 10 times faster than chromosome DNA, involving about 1 to 2 base changes in every 100 nucleotide for every million years, because it lacks the repairing enzymes like the nuclear um, chromosomes. Studying change in the base pairs of mitochondrial DNA allows us to detect evolutionary changes occurring over several hundreds of years before. During fertilization in humans, sperm contribute the nucleus to the ovum, egg cell. No mixing of mitochondrial DNA from a sperm with the egg cell. Hence, egg cell mitochondrial DNA is present in the zygote. So, the mother's mitochondrial DNA is passed to the rest of the generation. Evidence of evolution can be studied with the help of mitochondrial DNA and relationship between the organisms. Mitochondrial DNA, why it is very important for we have to study. Most DNA is located in the cell nucleus where it is called nuclear DNA 99%. Only 1% is accounted for the mitochondrial DNA. Small amount of DNA can also be found in the mitochondria where it is only 1%. Mitochondrial DNA is a circular, very short compared to the nuclear DNA. Cells contain any number of mitochondrial DNA ranges from few hundreds to thousands. The most part of mitochondrial DNA is inherited from the mother in a sexually reproducing species. In humans, mitochondrial DNA is nearly 16,569 base pairs long and consists of 37 genes and a control region which is called the not-coding region since it doesn't code for any gene products. It is called as introns. Mutation occur at a steady rate in all DNA, but nuclear DNA has its with this enzyme that may repair the changes. In some cases, these enzymes are absent from the mitochondrial DNA. Last slide. What we are going to see is what are the factors affecting the molecular clock, especially in the mitochondrial DNA. Different genes or proteins may change at different rate. Hemoglobin mutates more rapidly than cytochrome C. The rate of change for a particular gene may differ between different group of organisms. Over a long period, earlier changes may be reversed by later changes, potentially confounding the accuracy of prediction. That means, over a long period of time, earlier changes already changed DNA may again go for mutation, so it will create a confusion whether it is a new mutation or an old mutation. These are all the references I have referred for this. At last, I refer the Book of Biology, Cambridge International AS and A level.